Okay, I guess we'll get started. So welcome everyone to kind of the second half of the Microbiome Informatics webinar series. Today, well, my name is Benjamin Bolduc. I'm a research scientist at OSU, and I'll be basically giving you a huge overview of all of the available virus tools across the various cyber infrastructures, web platforms, analysis platforms, etc. I have quite a bit to cover, so if you have any questions, just feel free to ask them in the little Q&A feature. Yeah, and if those don't get answered, then I'll try to answer them towards the end of this. Okay, so I am right here in our little webinar series. Last week, Michaela and crap, I forget his name. Sorry.
Rory.
Rory, thank you. I can never pronounce his name. Went over DRAM and basically annotating microbial genomes. And so the real question I have for this and kind of to pose to you, or you're kind of posing, is that you have virus data. Now, what do you do with it? And so there's two real questions that I'm going to try to address. The first is what you can do with that data, and then where you can do it. So what you can do is kind of the typical, I want to assemble it, I want to get some population ecology, I want to be able to identify my viruses, I want to be able to identify the taxonomy and classify it and all of that. And then the other is where you can do it. And these are various resources that I'm going to go over. So hopefully by the end of this, you'll have a better understanding of what each of these kind of services provide. And they span cyber infrastructures, platforms, analysis frameworks, high performance computing, and kind of other resources. And they kind of do a lot. And each one of these itself has been a multi-day long workshop and session. So I'm not going to be able to do any of these justice. So feel free to go to these websites when we get to them. There's, entire series devoted to learning and working on each one of these. And I don't have enough time in these sessions to go over them with you. Okay, so this is the kind of the viral ecogenomics pipeline that I'm going to use as a basis for kind of orienting us on our journey today. As you can see, we can start with the quality control section. We assemble our data, we assess the quality, we identify viruses, and then we have this giant little section where we basically do what I call like the more real science, the more exciting stuff. The identifying the genes, annotating them taxonomically, identifying the various populations in there and what are they doing. And just to remind everyone, I'm going to go over the what. So that's in the little red and blue box here. These are the tools. Some of these tools that I'll be going over, I'll just handpick a few of them just to give you an idea of what that kind of section is capable of. And then next week, I guess you're stuck with me, is going to be actually how to do that. So we're going to go through and process a viral metagenome on KBase and on iVirus using resources available through iVirus.
Ben?
Yes.
Are you sharing your screen?
Yes, I should be. I apologize. Let me end my show. Okay. Let's see. Let's actually just share my screen. I wonder why it's not capturing it. Okay, apologies for that. Okay, can you at least see PowerPoint now?
Yeah, that's good.
Okay, and I'm gonna make it big screen again. Can you still see my screen?
Looks good.
Okay, apologies for that. Okay, okay, so I have virus data, what you can do with it, and where. And these are basically the areas that you can process. You can do some of this processing. And I've gone over that these encompass cyber infrastructures, platforms, analysis frameworks, HPCs, and other resources. And in the red blocks are kind of what we're doing. And in the blue box are like what tool is performing that kind of function. Like what is identifying our viruses, what is assembling our data. And on the outside is basically where we'll be doing that. So catching up where, so next week, we will be going through this whole process on both KBase and on iVirus using resources provided through iVirus. So the goals this week are basically to go over those tools to analyze a viral metagenome, what platforms are available to run these tools, and then third, how to decide on which platform is best for you and your data. Okay, so the first thing I'm going to say is that a couple weeks ago, Dylan went over MAGs, and pretty much these whole 6 weeks plus have been looking at everything from a microbial perspective. And I'm that token virus guy who's telling you that the whole world isn't just microbes, and viruses aren't microbes. And that really kind of impacts certain aspects of the kind of this ecogenomics pipeline. Some elements of the analyzing viruses is the same as you would a microbe, but in other stages and steps and parameters you might use are totally different. So I'm gonna try to ask, the question really we shouldn't be asking is like, why do we study viruses? Because that in and of itself is several courses, and you're probably not here for me to explain to you why viruses are the most awesome things to study. Instead, for this session and for next week, the question is actually, how are they different? What do you have to do differently versus processing a microbial metagenome? Oh yes, I went over this. Dylan went over a couple weeks ago, how to go through most of this pipeline. And so this is kind of like an interesting way of looking at, microbial data. So the first thing is that viruses don't have universal microgenes. And this is really critical because a lot of the way microbial informatics kind of orients itself is that they focus on the 16S. They focus on a set of genes that helps, tell them, oh, these are the functions of my microbe. These are the products of various processing metabolomes. So they're harder to study both in evolution and ecology because you just don't have those references often. And because they're viruses, they can't live by themselves. And so it's often hard to culture their hosts. I've seen estimates like 97 to 99% of microbial or viruses hosts can't be cultured. And if you can't culture the host for a particular virus or group of viruses, then you can't generate enough material to do certain types of analyses that you could casually do with microbes. And then because we're studying viruses, we often think of them through the eyes of their hosts. So what do viruses do to affect their hosts? And by extension, how do those hosts affect their environments? So kind of the two big things that people always talk about is that you might be looking at a microbe that's kind of really important ecologically. Like maybe it produces, half the oxygen that we breathe. So viruses killing them kind of has a huge impact on, how we breathe. And then you might have various nitrogens, sulfur, nitrogen, carbon cycling that are, regulated through microbial processes. And if viruses are killing them, that's going to affect at the global scale. So I think last time I gave this webinar, I discussed a little bit about how metagenomics can address these problems, but that kind of creates some problems of their own, as you'll see throughout this talk. And so, like I said, viruses aren't microbes. So there's some information from Dylan's talk that we don't need, like we won't need to do binning. We don't have to do, bin quality control. We don't have to consolidate those bins. There's a lot of analyses that we just can't do anymore because we don't have those universal marker genes. And pathways, I don't want to say forget about them, but when you're looking at it from the perspective of a virus, it's basically what specific microbial pathways can I affect in order to, make more of myself. So iVirus is a project that, some shameless plug here, that I am the lead developer of. We have 2 papers out, and basically iVirus is an effort to provide apps or tools, data sets, viral data sets, and documentation and protocols to the community so that they can analyze viral data on iVirus, and most recently, we published this just last year on KBase. So if you go to iVirus, you go to KBase, you'll be able to process viral data sets thanks to the efforts of iVirus and those iVirus partners that have contributed to those resources. The first I wanted to point out is protocols.io. iVirus uses protocols.io. Lots of different labs use protocols.io. If you're not familiar with it, you should check out the website. They offer a lot of different features that make it easy to publish your protocols, to get citations for your protocols, to navigate to other labs and see what kind of protocols that they're using in their research. And iVirus uses them extensively. I won't go over into too much detail what each of these provide, but for example, we have one protocol here, applying vContact, which I'm going to talk about, to viral sequences. You get a DOI for this. You get to look how many views and the user engagement. All the iVirus protocols, you can ask comments, and I will, time allowing, respond to your questions and concerns about the protocol and maybe how to optimize it for your particular protocol, for your particular step. There is great kind of step-by-step instructions that you can add to these. And I actually put in kind of these tips and author commentaries where they don't kind of fit in the traditional protocol setting. And so if you want to get the edge, you'll put in a little bit more effort in your protocol. You can try to change things up based on these tips and recommendations that I and other authors provide. And so you can join us at protocols.io groups/iVirus. Or if you just go to protocols and just search for iVirus, we should be one of the top hits, if not the only hit that you get. Okay, so this is, these are the two pipelines. I showed you the top one earlier. The first one is available on iVirus, and the second is on KBase. And so as I go through these various cyber infrastructures and platforms, keep in mind that you can't do everything on one resource. You can go through the whole pipeline individually on each of them, but you might not have, let's say, a specific assembler available on KBase that is available on iVirus. So definitely look through these protocols and kind of follow along to see which kind of infrastructure you want to use. Do you want to use iVirus? Do you want to use KBase? Do you just want to use your HPC? Before, investing all of your, eggs into one of these baskets. So the first thing we're going to talk about is quality control and assembly. Oh, okay, I was going through this. Okay, so the first step is, it's like I went before at the beginning, quality control, then assembly, and then assessing the quality to make sure that your assembly is good, identifying the virus, and then finally there's that large swath of various types of analysis you can perform. Okay, so I'm kind of going to group up what all of these tools, platforms, whatever, are trying to provide to you. They all provide compute power, so they give you CPU, CPU cycles, resource units, whatever you want to call them, in order to perform your analyses. These are all free with kind of a caveat that I'll explain later to perform whatever analyses that you want to do, provided they've got the apps or tools available to do so. They'll provide mechanisms of data transfer, so you can download data from the SRA, for example, to each of these resources. You can upload data from your computer to each of these resources, from your HPC to those resources, etc. Clearly, they have to provide storage. Each one of these provides storage in its own kind of way. And then they all have some sort of user interface. Even OSC, which many of you might be part of, has its own user interface if you don't want to use the terminal, and you can still perform various analyses, although a little less efficiently than if you were using a command line. Okay, so iVirus. iVirus is a NSF funded huge, I don't know what to call it, a huge behemoth. They have they've got kind of two different data centers. They're powering, I think this is out of date. I'm pretty sure this is out of date. I think it's 60 or 70,000 active users with thousands of different institutions that are partnering with them across the world. They're designed to provide kind of like a one-stop shop for all the different types of analyses that you could think about. They have thousands of different apps, many of which are not microbial specific. They're kind of just more generalized apps. Until cough, cough, iVirus came along, there were no virus apps here out of the thousands of apps that they had available. And these apps cover data conversions. So if you want to move from like FASTA to FASTQ, they have QC and data cleaners. And I say data cleaners because we're not just talking about, microbial or viral centric, read-based QC, lots of different assemblers, binning tools, annotations, sequence analysis, phylogenetic tools. The phylogenetics is far more extensive than we have time here to discuss, and 9 out of 10 of them, I have no idea what they do. They say stuff, oh, it does this type of Bayesian analysis, and There's a dozen of them, and each have their own kind of flavor of doing things. And then finally, there's terabytes of data sets available for you to use freely on iVirus. So they have a number of different products available to the end user, to you and to me. And then they've got a whole bunch of other stuff that is basically focused for the developers or those that want to leverage iVirus to run their resources. I'm only going to talk about the discovery environment and the data commons because one, they're the most relevant to the talk today. And two, they have week-long seminar series on all of iVirus's resources. And we don't need to talk about all of those other stuff. So I have a little video, let's hopefully this works, on creating an account and logging in. So hopefully my computer doesn't explode, and we will start. Of course, let's keep going. We're gonna start? No, maybe we're not gonna start. Okay, so I will manually move us along. So we can sign up. If you go to iVirus.org, you don't have to do that now. You can always come back to this talk afterward. And you can click on the sign up and then create an account. You can fill in your username, first name, last name, and your email. I think it does ask a little bit about your demographics, but you don't have to do that. You can put in whatever you want or not specified, and then go to next, and your account will eventually get created. It might take like a few minutes or a few hours, but eventually you'll get an email confirming that you've created an account, and then you'll be able to log on and use it. Okay. Oh, this works. So this is how to go into the Discovery Environment 1.0. It's going very fast. This is still available as a legacy resource, notifications, preferences. I don't know if it's always, I think you can access this like as a legacy, but by default, you'll have to hunt for it. It will default you to the 2.0, which I'll talk to in a second. They have data, apps, resources. I'm going to go into each one of these. So data, it's kind of a traditional file folder system. You have apps. You can look through all the high performance computing apps. There's, as I said, there's a lot of apps you can go through. One of the negatives is that there's so many apps. You have it doesn't orient you to where you are. So if you want to look for assembly apps, you can't just type in assembly and suddenly all the assembly apps pop up. You have to know what assembly apps you're looking for in order to find them and to run them. Analyses, of course, you have to have a way of organizing all of your analyses that gives you kind of details on what you've done. Of course, it's going to start over. Maybe it's because I already got to the end of this. Yes. And so this is kind of a very bare bones way of looking at on your data, the apps that are available and the analyses that you have performed. The 2.0 takes a more modern approach. It's 2.0, 2.0, there we go. And has definitely what you would kind of consider a more modern telling of the story. Let's see, sorry about that. Like the first, it has apps available that you can go in through. It has videos. Lots of different resources are available to you. The home is kind of the same. The data is still in the file and folder view. It's just a little bit more clicky. You have to go through. Apps is still the same kind of situation where you have to go through and look for what you're looking for and find specifically what you're looking for. And then, of course, you have the apps, you know, that you can look through, or your analyses that you can look through and kind of click on. They also have teams and stuff like that you can go through. And then finally, I think I have one more thing here. Oh yeah, if you ever need help, there's always a great help menu. iVirus is pretty responsive to most of your questions and concerns that you might have. So if you have any of those, just send them a short email. They'll actually have pop up like a little chat menu here for you. I'm not showing that here, but it's kind of what you can, anytime you go to a website trying to buy something, and it's like, hey, I see you're looking at, a new shirt. Let me help you purchase that. The same kind of process, although without the kind of trying to sell you something, is also available on iVirus. Okay, so iVirus is going through a transformation. So it's still funded by NSF. They still offer free services. But if you want things to go faster, you want to have more data available to you to store, how many jobs you can run at any one time, it's now going to cost some money because NSF isn't going to fund your huge resource forever at very large levels of funding. And so iVirus is defraying some of that cost, for better, for worse, on users. And so you can still use iVirus as a free resource to do your analyses, but keep in mind that the resources are going to be more limited for the free users than for those that are paying more, which is kind of a little nerve-wracking, but it's, it's a huge behemoth, like I was saying before. It has a lot of moving parts, and there's a lot of data availability and resources, available through iVirus. So it costs money to run. So you can't really blame them for trying to defray their costs. Okay, KBase. KBase is an alternative. Some people see it as a competitor. It's funded by the DOE, and their goal is to basically look at data analysis through kind of an interconnected web of data provenance and data relationships. Their goal is basically to connect to various resources, extract metadata, to be able to create what are called narratives to basically organize your experiments. And we'll go over what narratives are kind of in a second. And suffice it to say is that, I don't think I have it written here, but KBase is much smaller in scope than iVirus. So iVirus was, let's try to do everything all at once. Let's try to provide as much as possible in each one of these areas. And just the users are going to use what they want to use. We'll provide all the resources available to them. KBase, on the other hand, is far more directed. It's designed basically for microbial, viral, plant biologists, etc. Probably not giving it praise, but they're basically have a far more focused effort. There's fewer apps, fewer resources, fewer of a lot of comparisons versus iVirus, but they do so in a much more streamlined fashion. And it offers a lot more guidance and still leverages, lots of great apps, lots of great tools and resources in a way that many people might find a better experience to use than iVirus. I don't want to knock either of these, so don't try to take any subtle tones or whatever from me as like this one's better than this other one. I use both of them. I develop on both of them. I've spent years working with developers from both sides, both groups. No, like no bias here, hopefully. So I'll go through this whole process again. I'm not sure why it doesn't do that. Anyway, that'll remind me not to make movies in my presentations. And so you can go to kbase.us, sign up. It's the same process. Try to go through this more quickly because I'm using up a lot of time. You can sign up through a Google account or an org ID. If you're all fancy, type in the information. Don't have to, spoon feed this to you. You can actually access that through your institution, or if you have your org ID done through Google or Facebook, you can, log in through there. As additional providers, if you're an old person like me, once you could only sign up through Globus, because Globus is the data kind of support for iVirus. For KBase. It isn't, highlighted nowadays because org ID in Google is probably more often used feature for users, but if you have Globus, it still works. Okay. And okay, so once you've actually, the second video always plays. So you can log in, and what you have are narratives. And narratives are basically lab notebooks. I'll get to that in a second. You have various narratives. You can have those that are shared with you, your own narratives. They have tutorial narratives. They have public narratives. All the tutorial narratives are shared. They're all public, but they kind of break it down and kind of make sure that these are kind of the best narratives to basically to use as a tutorial instead of going public and looking through and trying to find them. Public, there's lots of different public narratives that can be shared. There's organizations that you can be a part of. I'm part of a couple of them. There's an app catalog that actually organizes them. I know this is kind of embarrassing to iVirus, organizes them thematically. So if you're interested in genome annotation, you can look through, and these are the different genome annotation apps that you can use. They have assembly, reprocessing. You can organize them by run counts. What are they called? Different categories. They have different versions. And so this is actually really great. You can look at the apps that are in development. So you can't really run those on proper KBase, but you can look at them. Beta apps, released apps. So beta apps are available to the public to test. And there's no guarantee that the app is going to work, but better chance that it is going to work. It might not be a fully finalized version of that app, but it should work. And then finally, you have the released apps. Those are the KBase validated, KBase verified. They've gone through a whole committee. I won't go into that. The released apps should work, and if they don't, the developer will cry. But you can sort through this whole list to see, what's in development, what is in beta. So if I talk about an app that you're like, hey, I'm not finding it, it could be just in a beta stage of release, and you can use it. It probably works. So if you do go to KBase hearing about an app that's being run, used, talked about, it might not be fully released because that KBase stamp of approval in the released means that it's gone through a lot of checks. Status, add modules. You can search through, this is kind of great. You can search through data that's available throughout all of KBase for specific kind of data sets. You can also search through JGI. So if you're a fan of JGI, or regardless of whatever you're a fan of JGI, if JGI has data for you that's searchable through JGI, you can actually do so. So my PI, Matt Sullivan, I type in, he's got some data sets available through JGI. You can click through, and then you can actually copy it, pull it over to your narrative, pull it over to your kind of lab notebook, your experiment. We'll copy it over. You can look through jobs, just like Cyrus had the analysis. I don't have any jobs running at the moment. So let's take a look at the narrative. It usually is that fast. And once you get to the narrative, I think I've got a couple slides after this that basically walk us through this whole process, but hopefully this guide kind of shows you that. On the left here, we have the data. So all the different data objects or properties. So these are the inputs of and outputs of any of the analyses that you run, any of the apps or tools. They're available in that kind of data panel. The apps have an app section. If you just quickly summarized all of those, you get to like a couple hundred versus the thousands of iVirus. But here you actually can, they're pretty well organized. You can go through and click on expression, read processing, fact, read processing, yes. We have BFC, we have FAFSQC, and then there's a lot of other KBase utilities. And I'm not going to go over the pros and cons really of quirks with KBase, but KBase needs to have things in kind of a certain form or structure. And most apps don't natively work in those data structures. And so KBase provides kind of like connective tissue to basically get you from one app over to another app. So if the first app requires FAFS, generates FAFSQ files with quality associated with the sequence, but the next tool requires FAFSA files with or FAFSA files without those quality, and it can't consume FAFSQ files. KBase has kind of helper apps that will make those conversions for you, or will convert from one object, like a reads object, into maybe a reads set object. Okay, look through some of the apps. iVirus, there's actually more. I actually took this like a year ago. There should be 4 virus apps there. All thanks to iVirus for providing those. Oh yeah, this is what I was showing. Yeah, so there's a Viromatcher, which I'll talk about. It's a virus host tool that was actually in beta development at the time. And so if you don't see it, it's released now. But if you don't see something, try to click on the little beta release flag and you'll see them. Yes. And so this narrative is basically a Jupyter notebook for those of you familiar with those. And this is a kind of a webinar that I presented for KBase about a year ago at this point, a year and a half ago, where I basically go through, and this is what we'll be doing next week. We'll be going through the entire process of processing a viral data set from end to end. I go through and I put like the background and the experimental design and stuff like that, just so that the end user, can see kind of the scientific background behind all of this. Yes, so I don't go through this whole process, but this is something that, like I said, we'll be going through. Okay. Gone through the data section, there's that app section, and then finally the narrative section that we just went through. And then, of course, there's extensive search features that kind of already went through. Okay, so I'm going to try to pick up the pace a little bit. So IMG VR is actually a viral resource. It's part of the JGI. It's their IMG resource that has kind of been filtered and curated down to only include virus-centric data. And there's a ton of virus data here. They have lots of different viral data sets, UVIGs, which I'll go into in a moment, viral OTUs, not to be confused with the 16S OTUs. They have these UVIGs associated with hosts. They have them associated with certain qualities. So if you want to be guaranteed that this is a particular virus, you can look at high quality viral genomes or fragments. You can look at mid quality. You could look at just any quality. Oh yeah, that's what I did here. And so I'll go into the UVIG standards in a moment, but basically these standards are a kind of a community perspective, a set of guidelines so that everyone in the viral community kind of is talking about the same thing when they talk about like a viral OTU or viral species or viral contig, like what quality that is. The pro and con of IMG VR is that they do all the processing on their end. So if you don't like how something was done, that's unfortunate. If you don't like how they assembled something or the parameters they used in their assembly, you're stuck with the JGI pipeline. But if you're happy with it, then you have a lot of kind of viral data that's available for you to use. I think I take us through this process. This is the first one, so hopefully this works. Let's scroll through a little faster. Oops, a little too fast. So you can click on one of those options and you can look through all of the VOTUs that are available in that particular section that I clicked on. They've gone through and kind of annotated each of the viruses, provided that they've got some hallmark gene that they can use in order to identify their taxonomy. Let's go through what OTU they're a part of, how they identified that, viral OTU, what kind of quality, the length of them, et cetera, how complete they estimate that the viral genome is, if they have any tRNAs that they found, how did they do this identification, how they check the quality, which we're going to go to, and that's check V, the ecosystem that it was found in. So IMG doesn't really provide any analysis per se, because it's already been done for you. It's already been pre-computed for you. So I find IMG VR is a great resource to like pull metadata down, pull like curated data sets that are associated with a particular host or a particular environment to then do subsequent analyses on a different in iVirus or in KBase or on your local HPC. Predicted hosts, if that's available, and how they've predicted the host. You can also search and filter through, of course, it's JGI. You can do a lot of filtering and sorting based on metadata and other attributes. So if you wanted to look for only soil VOTUs, you could actually group them by their ecosystem and then download that particular set of VOTUs. You can also drill down and get into more detail. I don't want to belabor the point, but if there's data associated with it, JGI is going to make that data available for you to kind of access or look at, et cetera. And these are all the different aspects that we were looking through earlier. And as I said, they've already done all of these analysis. So you can actually look through and look at the genome annotations for each of them based on KEGG or COG or PFAM, TigerFAM, et cetera. And you can actually, I don't know if I click on them, but you can actually look through, I'm going through the different genes. It's a hypothetical protein. You can actually search through and filter for other genomes that might have that same gene. And then I think I have it here. Yep, okay, you can. Yep. So you can filter through and find other VOTUs or PFAMs that they might be associated with, of course. And then, of course, you can click on them individually. You can add them to your cart. You can actually click on whole groups if you want. You don't necessarily have to do them individually. I think that was the end of that one. Okay. So we're about 45 or so minutes in. So these are the two pipelines that, just to everyone to reorient themselves, take a breather. So we've gone over very briefly iVirus, KBase, the resources available at IMG, VR, and those are kind of the 3 different flavors, basically. So now what we're going to do is we're going to go through these and just talk about what each kind of stage or step is available, and like what you should be thinking about at each one of these stages. So the first is going to be QC and assembly. That's the kind of front end of our pipeline. And I know Dylan went over a little bit about read quality control and assembly. There isn't too much different in terms of the tools that you use to read clean reads or to assemble. But a paper was released a couple of years ago by Simon Rue, who's over at the JGI now, that basically looked at this from the viral perspective. And here you see, we won't go into too much detail, but on the y-axis, we're looking at the percent of genome that was recovered. On the x-axis, we're looking at how much of the genome was covered in this particular data set. And so basically, to orient yourself as you're moving from left to right is, as you add more data to your assembly, do you get better recovery of your genomes? And as you would imagine, the more data you add from having only 1 or 2x or 3x coverage, you recover a greater percentage of your genome that you've kind of put in there. And so what Simon did here was look at various different ways of cleaning up your reads, and basically he did this for a various different number of assemblers, but basically to summarize, just looking at Metaspades, for example, he found that it doesn't really matter what you use to clean up your reads, as long as you clean them up. So clean up your reads and you add more sequence data, and it doesn't matter, really. You can use K-based tools, you can use Trimmomatic, you can use digital normalization tools, etc. It doesn't really matter because, coverage in this case matters far more, and cleaning up your reads matters far more than how you're choosing to clean your reads. On iVirus, there's a whole bunch of tools available. This is a short snippet. These are basically the ones I think that most people would be familiar with. On KBase, the list is a little bit shorter, but no less effective. It doesn't matter if you're using BB tools to clean your reads or Trimmomatic. The fact that you've cleaned them up makes far more of a difference. The next thing Simon went through is to look at what is the best assembler. I know that can be pretty controversial, and I'm going to tell you it really doesn't matter. Now, there are certain, I would give the edge to certain assemblers in certain situations, which is basically what I'm going to go through now. So if you look at that kind of same type of process, basically with what kind of recovery you can see on the y-axis and how much coverage you have on your x-axis, you can find that almost all of the assemblers perform IDBAUD, MetaHit, sort of MetaVelvet, Omega, Metaspades. They all perform better the more input data, the more coverage you give your genomes. And I know that sounds kind of obvious, that the more data you give, up to a certain point, mind you, the better your assemblies are going to be. But you kind of have to prove that. You have to show that. And Simon I think very cleanly in this particular, benchmarking data set showed that. I would say MetaVelvet is kind of fallen out of favor. I would probably favor Metaspades, Omega, Megahits, and even to a lesser extent, IDBA UD over MetaVelvet. But there could be certain situations in which MetaVelvet performs equally or better than some of the other assemblers shown here. The next thing I wanted to say is that certain assemblers have certain advantages and disadvantages, and this is what I was getting to a little earlier. So if you're looking for chimeric contigs, if you want to reduce the amount of chimeric contigs, and what is a chimeric contig, you ask? A chimeric contig is when you have two or more similar but different genomes assemble into the same sequence, into the same contig. So let's say you have two different strains, I shouldn't say strange, maybe two different species of viruses that share 50%. This can be common, 50 to 70% of their genome. Some assemblers get confused by a lot of those very similar sequences and overlaps from these two very separate viruses. And so they put them together into the same contig. And in a perfect world, we don't want that to happen. And if you notice, Megahit has a slightly reduced chance of that happening. So the percentage of chimeric contigs overall, and then of course in the 10 KB or larger ones, is reduced in Megahit over, let's say, Metaspades or IDBA UD. So if you're working in an environment where you might identify a lot of chimeric contigs, or you're like, I don't want chimeric contigs at all, you can choose Megahit, and it has a slight advantage, at least in this test case, over IDBA UD and Metaspades. However, if you want to look at false positive circular contigs, so one of these assemblers like to do is it will often get rid of a lot of the kind of repeats or other kind of ends, like poly A tails, for example, on the ends of your viral genomes or your contigs. And so that can cause an artificial circularization, basically, of some of your genomes. And the problem with that is if you assume that if a genome is circular, that it should be complete or should be closed, or, but that might not be the case. Or if it does circularize, it might not be, a true virus. And so if you're looking to reduce the number of false positive circular contigs you're going to find, Metaspades is actually much better than Megahit IDBA UD. And so, like I was saying, each assembler has its kind of own advantage or disadvantage when it comes to assembling your particular data set. And it might really matter what environment you're in. Maybe you're in a particular environment that favors chimeric contigs, or maybe you have a lot of small, short, circular genomes. And maybe you want to make sure you reduce the number of false positive, small, short circular genomes that you have. So you might choose Metaspades versus Megahit, etc. But one of the biggest impactors, basically universally, is strain heterogeneity. The more, I would say, the more similar sequences you have in your data set, the harder it's going to be for your assembler to put those genomes together. And that is universally true. When you're working with microbial data, this is also important, but it doesn't have the kind of outrageous impact that it will in a viral data set. And so a lot of these assemblers, basically because of the assembly algorithms, they're very, very sensitive to strain heterogeneity. So at all costs, you want to try to reduce that in your data set in order to prevent, or in order to get the kind of the best or most efficient assembly. Yes, okay. So that was my note to me to not talk too much about that. So to summarize, clean your reads. Doesn't really matter how you do, just clean them. More coverage up to a point gives you better assemblies. You can choose whatever assembly you want to use, but they each have their own strength and weakness. However, it really doesn't matter. If you're looking for like a 1 to 2 to 5% improvement, that's not going to happen. You can try to switch between them, and you might see better with one versus the other, but that's not going to, really affect the overall outcome of your analysis. In practice, I probably should have written this here. I generally go with Metaspades as a first pass. And if Metaspades, which is known for consuming all of your memory and all of your CPUs, hits memory limits or wall time limits, then I simply switch to MegaHit. Just because it has reduced kind of resource requirements. The next thing I wanted to touch upon is viral identification. So we've gone through basically the whole quality control assembly. We've assessed the quality. We know that the assemblies at least looked good. Now we want to identify the viruses. However, we don't have any marker genes, which is kind of a problem. We don't have the same advantage that we do in the microbial space, where we can look at 16S genes, or we can look at certain other housekeeping genes that we can use to kind of bring together our genomes or bin them together into a mostly single cohesive unit. But thankfully, we have a couple tools that's not so much of a problem. So virus tools have identification tools have kind of been broadly organized into two major categories. And those are the gene content and genomic features, which I'm going to go through in a second, and basically looking at their k-mer frequencies based on a particular training set. So the gene content and the genomic features, they're kind of like the typical first principles. What does a virus look like? What kind of hallmark genes do you have? Do you have a terminase? Do you have a major capsid protein? Do you have a portal protein? Microbes don't have these proteins. And so if you find these particular genes, identify these genes on a particular genome, then you can be more or less confident that that is a viral genome, that is not a microbial genome. Of course, there's some caveats to that, but in general, if you find a particular genome, let's case in this particular, for genome 1 here, it has a terminase large subunit, major capsid protein, and a portal protein. That is almost guaranteed to be a virus. And then also looking at genomic features like strand switching, reduced strand switching, because if you look at a microbial genome, you notice that the genes can go, forward direction, reverse, forward, and they're all different frames, and they switch up repeatedly as you just kind of visually scan through the genome structure or the organization. And viruses don't always do that. They do it less frequently. Of course, in the virus world, there's always a virus that is an exception, or always a virus group that's an exception. But broadly, generally, viruses don't strand switch as much. They have shorter genes than microbes, and it's kind of weird to say, but they have reduced PFAM hits. So the fact that you don't hit anything can be a sign of a viral nature. And so you can design tools using the gene content and genomic features. And then beyond that, to kind of give you the flavor, so you have these two categories, major like philosophies of thinking. But then you can either apply statistical methods or machine learning or both of them to each of them. So you can imagine if you have gene content, you can use statistical methods based on that, like, hey, I've got 5 hallmark genes. And so the chance of this being a virus is 100%. I've got 4 hallmark genes, the chances being 80%. I've got 3, etc. You could do something like that. Or you can use machine learning to say that, hey, I've got these set of genes that have lots of hallmark genes, and they've got these particular genome organization characteristics. Let's throw it into machine learning and have that figure things out for me. So the first two that I will talk about is VirSorter. So VirSorter 1, published a number of years ago, it was kind of the gold standard for a number of years. Some people might still consider it the gold standard. It basically used those gene features and used a combination of statistical methods, actually a probabilistic model in this case, to basically look at the features and ask the question, do you look viral? And then VirSorter 2 came along last year, and that uses multiple machine learning classifiers. So it kind of combines that gene content and genomic features with machine learning. And many of those features, like the hallmark genes, the percent of viral genes, many of them were shared between VirSorter 1 and VirSorter 2. And you also notice that VirSorter 2 uses a lot of other kind of features that VirSorter 1 didn't. And of course, using machine learning, you can see improvements. I don't know if I put the improvements in this slide. No, I didn't. But clearly, VirSorter 2 performs much better than VirSorter 1. The second tool is Marvel. And Marvel is very similar to VirSorter 1. It has many of the same kind of features selected, gene content and stuff, that VirSorter 1 used. But instead of using statistical methods like VirSorter 1, it uses machine learning instead. And arguably, Marvel performed equal to or better than VirSorter 1, of course, at the time of publication, which kind of harkens back to kind of a problem with a lot of kind of viral identification tools, is that they're always comparing it to like the older models or their older versions of some of these tools. And so, of course, you see a new viral identification tool and it's like, hey, we perform better than, you know, this tool and this tool and this tool. But you really have to look into and delve into like how, like, what length of genome is this best at? What kinds of genomes does it perform better at? Does it perform better at DNA viruses, RNA viruses, human viruses, et cetera? So always be kind of conscientious of what tools are being compared against what other tools and measuring its kind of metrics. And then finally, we have Deep Virofinder, which is pure machine learning based on KBase frequencies. I won't go into details here, but basically, they fed basically a whole bunch of viruses, known viruses, into various machine learning algorithms, identified which KBase frequencies were most indicative of being able to differentiate between a virus and a non-virus, and basically built a model for that. Yes, I think that's all I had that here. And I do apologize that I can't go into super detail about each one of these, but each of them, like, unfortunately, is like a whole hour talk, and you know, they all work. They all work. Just pick one, use it. I'm partial, of course, to VirSorter 2 and Marvel, mostly VirSorter 2 and also a little Deep Virofinder. I use Deep Virofinder for a lot of the smaller genomes. I should have probably put a slide for this, but a lot of the gene content, gene features, genomic features, whatever, they perform much better 5 kb and above. So if you have viral genomes that are, 1 kb, 2 kb, 3 kb, there's often not enough kind of genome or genomic content in order to make statistically confident predictions. However, a deep Virofinder trained on shorter KMER frequencies can identify largely successfully those genomes that are below 5 kb, 3 kb, etc. However, I generally don't trust things below 1 kb and even up to 3 kb, even predicted by Deep Virofinder, because there's a huge high false positive rate once you start getting into like that short of a length. Okay, so VirSorter 1 and 2 available on iVirus and KBase. We have protocols for running them. There is protocols for Marvel. I didn't put that here, but there is one for iVirus. And then Deep Virofinder also is available on iVirus. Yes. So if you're looking at quality, just kind of like take a screenshot of this. This is published on protocols as well. But each tool kind of has its own set of what should be considered high quality, medium quality, and just low quality. And it's really confusing when you're trying to run your data set on multiple different viral identification tools, and the qualities are different. So high quality for Marvel might be 90%, and Deep Virofinder might be greater than or around 0.9. And so you're like, oh, okay, maybe, they're kind of sort of similar, but then you go to something like VirSorter, which uses categories. And so if you wanted to compare VirSorter 1 and Marvel, then you're going to have to compare these categories on a, case-by-case basis. I've put the recommendations that we in the Sullivan Lab use to kind of distinguish between the different quality levels in order for us to kind of compare them. The lengths, I think I have Deep Virofinder 1.5, but really there's some caveats for that. Some of the pros of each of these, many of them are easy to use. VirSorter 1 and 2 are really easy to use. Deep Virofinder is very easy to use. Virofinder and VirSorter 2 are very fast versus, the original VirSorter 1. VirSorters and Marvel, because of how their algorithms are designed and how they function, are pretty time consuming. They're also a little harder to use because of that. I found that Marvel's scoring is a little harder to predict and interpret, mainly because you can have medium qualities that are like 80%, and that's a false positive, and then you can have something at 65%, which is a false negative. So that's virus. So once you start getting into the medium quality, as long as you don't have like hundreds of thousands, you should really kind of try to spot check and look at them kind of generally to see which ones look more viral versus which don't really look as much viral. The only other thing I want to highlight is that high scores for Deep Virofinder can be false positives. So please do look at, even if the p-value is 0.05 or 0.04999, please do look and inspect a lot of those viral genomes to ensure that they're really viruses. And then finally, VirSorter 2, just go on protocols.io, click on VirSorter 2. There's a huge SOP that answers all the questions that you can possibly want to ask about VirSorter 2 and what kind of quality of your data set that you're getting. And then, of course, I bet the links that I would start trusting the the predictions. Now that's not 100% guaranteed. I'm not guaranteeing you anything. But this is kind of the gray area where I'm like, okay, now I'm going to start looking at things to have confidence in. Okay, and now there's a handful of different tools that I wanted to touch on. I'm actually making really, really good time. I think I might have cut out too much. When I did this earlier, I went way past 2 hours, and I didn't want to keep everyone here past 2 hours. Okay, so this is kind of like you've identified your viruses, like now what can you do with them? And up until a few years ago, we basically had no idea. You basically had just a few tools, and then you would basically apply microbial type analyses to your viral data sets. But now we're in the modern era, we're out of the dark ages, and we actually have tools that are designed to work on viruses. Before that, the question always comes up, should we be removing duplicates or very similar sequences? And if you follow the MuVigs protocols and guidelines, yes, you should. You should go through and remove or cluster any of your sequences that are at 95% ANI, average nucleotide identity, over at least 85% of their length. I think that's a little, yeah, that's a good starting point. Those are the minimum standards that you should have. I think in the lab, yes, okay, let's keep it there. So on iVirus, if you want to remove duplicates, I've written just an in-house script that the Sullivan Lab's been using and put it on iVirus called cluster genomes. You can do this locally too. Like I'm not stopping you if you want to run your analyses on iVirus or KBase, download the data, do something on your local HPC, and then upload it back to do later analyses. Deduplicate it, throw it into any tool that you want for, clustering or deduplication software that 95% ANI, 85% length, or something approximately that, and you should be pretty good. For cluster genomes, we recommend 95 over 80% length. In practice, you're not really going to gain or lose too many with that difference of 5% over the length. If you actually look at the kind of the coverage difference in that 5%, it's often negligible, and it's something that you don't really have to worry too much about. But basically that 95% ANI and over some large majority of the length is really all you need in order to get kind of pretty high quality genomes or contigs. This isn't on iVirus or KBase, unfortunately, but this is a tool that I've used frequently, DREP. I know a lot of microbial people are using DREP to deduplicate their genomes. I've used this, I actually use this sometimes more than cluster genomes on my local machine. And you can put in the parameters 95% over 80 or over 85%. But I strongly recommend for all the tools that I'm about to talk about, you should cluster your genomes or deduplicate them beforehand. Okay, I'm going to go into this. So after you dereplicate your genomes, you can process it through CheckV. And CheckV goes through a number of different processes. I love CheckV. Please do not publish any viral data without running it through CheckV first. Because CheckV does a lot of, okay, actually talk about it. One, it removes host contamination. It uses a set of microgenes and various microdatabases, viral marker, gene sets, databases, in order to identify viral marker genes. And Not every virus has a microgene. CheckV knows this, but more often than not, there's several microgenes or several elements of viral hallmarks in each predicted contig, each predicted genome. CheckV capitalizes on that aspect. And because of those microgenes, it can basically predict, you have, viral microgene. Oh, microbial processing stuff. Oh, 16S gene. Oh, other microbe stuff. It's like, okay, so where do we stop seeing stuff that look viral and where do we start seeing things that look microbial? CheckV basically identifies those boundaries, those junctions, and collects them. It also, because of those microgenes, can actually estimate the completeness. So let's say you have, I don't know, some virus, maybe some coronavirus, and it has 7 characteristic genes. And you identified a whole bunch of genomes, and they've got 6 of those 7 genes. So actually, let's not do that because I can't do that math. Let's say there's 10 genes and 7 of them pop up. Let's say that's a 70% completion rate, or they estimate the genome to be 70% complete. Now, this isn't perfect, but it does get you broadly to some meaningful measure of completion. And if you have high confidence, you might have 70%, medium confidence, 50%, because in practice, as exceptional as viruses are, they usually have a couple kind of viral hallmarky-esque genes. And if you've mostly assembled them, they should mostly include those genes. It can also identify the ends, assuming your assembler hasn't destroyed them. And this is especially true if you have direct terminal repeats or inverted terminal repeats. Harken back to, classical virology, and packaging mechanisms and all of that. But you can use those to not only estimate genome completeness, because if you have most of your hallmark genes and you've got 2 direct terminal repeats at your ends, you probably have a pretty complete genome. And you can also use those to make sure that you're actually at the end of your genome. And then finally, it summarizes the overall quality of what it thinks your genome is. And then, of course, it uses this quality based on the MUVIG paper, which I think I've mentioned before, kind of a set of guidelines that you should use when publishing viral data. Available on iVirus and KBase, of course, and on your local HPC or whatever else if you want to download the tool. Okay, and so one of the big things that people love to do with viral sequence data, or with any data, in fact, is basically try to estimate their abundance in various data sets. And in that same paper that I discussed before, Simon actually went through and basically estimated what threshold should you use in order to be able to confidently predict that a particular genome was there. And I can basically summarize this whole data set. Maybe I could have just put one figure here. And basically, you can look at this threshold, or actually, you could look at pretty much any of these. Many of these basically show the same thing, is that once you hit about 75% identity and you cover about 80% of your genome, you can confidently, mostly confident, I don't want to say everything's concrete, but you can mostly confidently assign that genome as present in that particular sample. And the reason this is important is because before this kind of point, it was kind of the wild west. where some people would map at 95% identity or 97% identity across, 95% of their genome, and they'd find that, oh, their genome is only present in a few areas. Or they'd have totally permissive things just saying, hey, I'm going to throw this into BWA or BB map or any one of the read mapping tools. And, oh, look, I've got my genome spread across the entire globe. Do you really? And so Simon went through, basically identified that and said, okay, above this, you can be pretty confident. Below this, you shouldn't trust it, or not as high confidence. And so you can basically, use those kind of confidence metrics, confidence thresholds, and apply that to your various, your viral data that's been predicted, cleaned up, and all of that, and map using all of your reads from all of your different samples and data sets. On iVirus, I think I might say it's the next slide, but yes, on iVirus, I've written two tools called Bowtie Batch and Read to Ref Mapper. I'm not very good at naming things, so you can use Bowtie Batch if you have many, many different data sets, many, many different samples. So if you have a whole, a dozen read files, you can basically click on all of those different read files in iVirus and drag them over to the app Bowtie Batch. And that will figure things out, put them in the right order, pairing and whatnot, make sure they're going the reads are going in the right direction, et cetera. If you've interleaved them or if you've not, and basically run Bowtie on all of those data sets separately, and then combine those results. Read to Ref Mapper will take those reads and map them to the reference. And you can generate plots, not exactly like this, but you will get the heat map and tables, the heat map that represents basically this table. And I don't think I put the example in here, but I do have one that has like 6 or 7 of them. It doesn't look as kind of fancy or flashy as this because I only have 6 or 7 data sets and there's only like 10 genomes in it. But if you did have 42 or whatever metagenomes and you had, hundreds of viral genomes, then you'd be able to generate a figure almost exactly like this on iVirus. As an aside, I think it would probably be best for you to run this and then, you get this kind of image, but I would really take it, download that file and make something really pretty in R or in Python, just because this is a very bare bones visualization that I wrote into there, just simply because there's not really visualization tools in iVirus. I do have CoverM mentioned. Okay, yeah. So CoverM should be available on KBase. I think it still might be in the beta stage. So Bowtie Batch and Read to Ref Mapper basically use like a component of CoverM's predecessor, and the developers of that dependency updated it to be more expansive, and so they came out with CoverM, which basically supersedes Read to Ref Mapper and Bowtie Batch, but it doesn't really give you any better results, which is the reason I still talk about Bowtie Batch and Read to Ref Mapper. It does the same. They do the same thing, but CoverM is a little bit faster and it's a better overall package versus what's available on iVirus. They still do the same thing, still function the same, etc. But I do find that CoverM is faster and easier to use. And in fact, I bet Dylan, I think, talked about that. So CoverM, you can use it if you're on KBase. The next thing that we'll want to do is talk about taxonomically classifying viruses. And this is kind of a controversial topic. I really wish I could have talked about a couple of the other tools. There are only a couple other tools that you can use in environmental settings. But the problem here is because you don't have universal marker genes, you can really only classify viruses that are known. So yeah, of course, if you're working in, an influenza lab, or if you're looking in a, I don't know, a plant and animal genome lab, you know what your viruses kind of are going to look like. You know what their hallmark genes are, you know how they operate. And if you're running samples and you wanted to look at the distribution of this particular, plant virus in this crop, of course, you might be able to visually see it, but you could grab a bunch of samples and, process them, and you can look at particular genes, and of course, you'll be able to identify your virus of interest. But you can't necessarily do that in a totally novel environment, a novel situation. So you're sampling at the bottom of the ocean. You have no idea what you're going to see. You're sampling a new microbe, you're sampling a new, whatever organism. You might not know what hallmark genes are there to look for, if they even exist. And so vContact was a tool that's been developed. I'm one of the developers, co-lead developer, lead developer for this tool that basically identifies novel viruses at the genus level. And I won't go through this in too much detail, but basically it uses the fact that viruses that are similar to each other or taxonomically related to each other should share many of the same genes. So if you're influenza, you're probably sharing a lot of the same genes. If you're infecting, if you're the same group of viruses that are infecting similar hosts, you probably have the same or similar machinery to infect the same hosts, etc. So if you have two viruses and they share 9 out of their 10 genes, the odds are that they're taxonomically related. And so when I say odds are, there's actually a statistical metric, a statistical confidence associated with this prediction. And so we actually get genus level taxonomy on both genus level, clusters, basically. So we can basically group these novel viruses into groups that are related confidently at the genus level. And we've applied this to a variety of different environments. We've applied it to RefSeq, because of course everyone always uses RefSeq. And we've basically been able to get the vast majority of all viral genomes accurately predicted. I think we're at 94% on average. It spikes up to 95 or so, 96, depending on what environment you're working in. Or it can be a little lower than that, but on average, about 94% accuracy, which is actually pretty good when you're considering you could be looking at totally novel, totally naked viruses, which you have zero predicted, I should say zero known proteins that are predicted. So you can have literally 10 genes, all hypothetical, and we can have pretty good confidence that we can put it into the correct genus for you. There are a couple other tools out there that I don't really talk about here that do promote a higher percentage or higher accuracy. They're like 96, 97, 98, even in some cases 100%. But keep in mind, as a caveat, that those are known viruses. So if you have known viruses, next week, actually, I'll mention them. If you have known viruses, then yes, you can use those tools. You can use BLAST, or you can use those tools to identify those viruses. vContact 2 is designed to classify anything and everything in the virus. I should say prokaryotic. That is my one caveat myself. This can work for eukaryotic genomes. I have not used it personally in eukaryotic data sets, but when processing viral environmental data, there are eukaryotic viruses identified in there. And yes, they are classified at the genus level, but I don't want to say anything more than that because I don't want anyone to, this is recorded, so try to call me out. I cannot say that it will work for eukaryotic genomes. This is available on KBase and iVirus. This is 17,000 genomes clustered, identified with vContact. The red are the viral RefSeq genomes, and in gray are the little clusters of genus level taxonomically classified viruses. I wanted to say something else here. Oh yeah, so if you're using vContact 2, there is a limit. So I think the hard limit is somewhere around 750 to 800,000 genomes, which seems like a lot, but I get emails all the time to increase that limit. Yeah, use this for 10,000, 50,000, 100,000, 200,000 genomes. It should work, no problems. I do have some notes on the protocol about how to handle larger data sets, but do keep in mind that you can't just throw all of IMG VR at this. You can't throw millions of genomes into this. You'll have to split up your data or follow my tips and tricks on how to process them using vContact. So I won't talk about DRAM-V. I think this might be why I've saved some time too. DRAM-V, because we actually talked about it last week. DRAM-V is basically that that metabolic, those annotations for viruses. So once you have identified your viruses, you can taxonomically classify your viruses. You can find AMGs from your viruses, identify what kind of functions or properties your virus could have, and how it could be affecting their microbial hosts. And so if you have any questions about how DRAM or DRAM-V works, it's the same process. It's the same basic tool. Definitely refer to last week's webinar with Michaela and Rory. Sorry, I think I was thinking Mike. The paper has Michaela and Mike, and so I got confused. And so yeah, just look at that, and if you have any questions about that, it's really easy to use. KBase is actually really easy to use because you can actually select the VirSorter, the vContact 2 or the VirSorter 2 output, just click on it and it automatically figures everything out, and then you press run. And of course you get your beautiful figures at the end of it. It is not so quick and easy and convenient on iVirus, but it still functions the same way. You just have to, click on a few more drop downs and click on a few more, accepts or runs kind of things next. I think that's all I wanted to say about DRAM-V. Okay, and finally, Viromatcher. So this is an unpublished tool, although we do have a Bitbucket repository with this code. And this tool is actually used. So it's not published as a tool itself, but it has been used in the Gut Virome database, where it actually classified about 50% of the 33,000 viral populations. Yeah, I should explain what it is. So basically, it identifies the host for viruses in your metagenomic data set. It is a tool that basically leverages a variety of different tools. So CRISPR spacers, prophage, host tRNA genes. It uses KMER signatures provided by WISH, and it aggregates all of those results into kind of a cohesive framework where they basically The author, Ahmed Zayed, he's an author on the gut biome database paper. He basically realized that, okay, if I've got a blast match of this virus, like into this host, or I've seen this virus integrated into this host, I'm pretty sure that virus is, that microbe is its host. And so you should have a lot of confidence about that versus maybe a KMER signature where it's like, oh yeah, I have got, a single 12-mer hit or 16 single KMER hit between that virus and that potential microbial host. I'm going to give that much less confidence. I'm going to, assign a lot less confidence to that KMER than I would be for an integrated prophage or maybe a couple host tRNA genes versus maybe in the middle of the road, you've got a couple CRISPR spacers matching, but that's it. However, if you have a whole bunch of KMER signatures or you have a whole bunch of spacers matching, then of course, even if you have not a lot of confidence about a single one, you've got, let's say, 20 matches, then you have a lot more confidence into those two genomes, that virus being associated with that particular host. And this tool basically formalizes that whole process. It's something that's been used kind of in literature over the years. People have used prophages to identify hosts. People have used tRNA genes to identify hosts. They've mixed and matched a couple of them. This is one of the first-ish tools. I'm sure Ahmed would correct me, to basically integrate them all into like a tool, a framework to work with that basically put in your microbes and your viruses, and it will generate a table of predictions with how many spaces you have, how many prophages you have, et cetera, and then the overall weighting for each one of those and the overall score so that you can have confidence in that prediction. This is available in KBase, as you saw in that narrative earlier. It is released. It was beta when I gave this earlier, but now it's fully released. There are two other tools that I wanted to mention because I didn't want to just seem like, Viromatcher is the only tool you can use. There are two competitor, similar tools. There's iPop, developed by Simon Rue. I mentioned him a couple of times over at the JGI. This actually integrates everything that Viromatcher does, most of the tools that Viromatcher does too, as well as this tool called Rafa, which in and of itself is another tool that you can use to predict viruses, predict hosts for viruses. So basically iPop combines everything. And it, according to the author, is superior to everything. Of course, that's the way of all tools that come out. Their tools are always better than everything, or at least equal to everything else. And in our hands, Viromatcher and iPop basically perform very, very similarly, such that there might be like a little gray area where they're probably perform at the same level. But, wait for the paper for more concrete benchmarks. I think bioRxives has maybe the paper out, or maybe they're getting ready to publish it or something like that. But yeah, stay tuned for that. Okay, so with that, I think this is my last slide. And so this is the whole pipeline that we're going to be going over next week. I will go through most of iVirus in the context of protocols.io. So if you want to follow along, if you want to create an account, you can. I've got example data, example input and output, raw data sets, et cetera, available for you to use on iVirus as well as on KBase. I'll probably go through iVirus a little bit more quickly because it's basically the same steps as KBase, but KBase has more visually appealing look to it, at least in my perspective. And so it's a lot easier to kind of slow down and go through steps in detail, even though the same steps exist on iVirus. It's just a lot more raw, I guess, unfiltered on iVirus's end. I think that's it. Yes. Okay, so I'm going to exit my screen, my slideshow. I'm going to start addressing some of the Q&As. I do apologize at the beginning for that the issue with the sharing the screens and stuff. Let me go over to Zoom. Maybe. Question answer. Okay. So I'm going to go through and I'm going to, okay, so question, do you have any comment on using Diamond for viral identification? Actually, I use Diamond internally in vContact too, to help match and identify proteins. And so I have used Diamond to help with viral identification, but really in this case, I think the bigger question is which databases you're going to use Diamond against. Because yeah, Diamond, you know, if you have a database of hallmark genes and you're looking for a particular viral type, yeah, you could use Diamond to search through that database of those hallmark genes and, you know, help identify your viruses. I would argue that your better use of time, better use of your CPU cycles would be to use one of the viral identification tools that I mentioned, either Marvel, Visorter2, Deep Viewfinder, to identify viruses. But yeah, sure, you could use Diamond. It's a more classical way of looking for viruses in your data set. Or alternatively, I know that some people have done this. If you have really large data sets and you are looking for a specific type or group of viruses, you could use those viruses and Diamond to search through proteins in a very, very large database like IMG. I hope I answered that. A second question, if I didn't answer your question, definitely email me or ask another question. You mentioned that it's not required to bin viral contigs. I saw that there's a pipeline for binning viral contigs. Oh, what did you take? Oh, on Virime. Okay. I think Virime was published far more recently. So something I didn't want to cover too much because I made this mistake in one of the first virus webinars that I did, is that I tried to include like the cutting edge tools before they were more mature. And so we've taken a more conservative approach in iVirus to only put more benchmarked and tools that have been used by multiple labs, not just by me personally in the Sullivan lab, but by other labs around the world before basically trying to integrate them into iVirus and iVirus and putting all that development time into them. What is my take? Yeah, I don't want to flip-flop around this. The problem is that I haven't used it extensively enough in my own work. Yes, I've read it. Yes, I've installed it. Yes, I've run it on a couple data sets, but I haven't really explored it in the way that I should in order to give you a good answer. So I'm sorry that I can't really answer that. But on the philosophy of binning viral genomes together, it depends on what level you're binning them at. Like, are you talking about a viral genome, a viral species, a VOTU? And you need to be really clear and understand the terms that you're using, simply because in the viral sphere, viral literature, viral species, VOTUs, viral populations, they all mean really different things, or they can be very different things. And some people who don't know use the same, who are thinking the same thing, but using all of those terms. And so it's created a lot of confusion in literature. But yes, if you wanted to bin your viruses to a specific species or strain even, then yeah, you're free to do so. And I would, you could still use any of these tools. Yeah, you just have a different kind of unit for counting them, I guess. But yes, that's my long-winded answer. What quality should be published? That's a great question. I would follow the MUVIG pipeline and recommendations. I believe when you're, I can't remember the exact, but I believe it's medium confidence and above should be considered publishable. Like those are the ones that you should be doing your ecological analyses on. A lot of individuals will take a 500,000 predicted viral genomes and just submit them all without any type of quality information associated with them, without looking through and identifying, what are high confidence, low confidence, no confidence. And then labs like ours, myself and others included, will download those thinking that they're all great quality genomes. And we're like, hey, there's microbial sequences in here. Like this virus, this isn't even a virus. Which is why I wanted people to be to look through the Deep Finder findings, some of the Marvel confidences, and basically make sure that what you're putting out there to the community is at least medium quality, if not high quality. In our lab, we often run multiple virus prediction tools because they all have their strengths and weaknesses and areas that they're better or worse in. And then we aggregate and combine those to have a reduced, granted we have fewer viral genomes that we're talking about, but we have a lot more confidence in them. And we'd be rather putting out a lot of high quality, high confidence genomes to the community than just throw whatever we have out there. Okay. Another question. Virus level taxer created visual tools to use, and is there a script? There is some information on the protocols.io website. If you just type in vContact2, there is a guide for actually taking the results of vContact2 and generating that network that you saw. The first network that you saw. The second network, the one that has 16,000 genomes, there is a script that I use that I have been sharing with people one-on-one who email me because the script, even though it's public, I should find where one of the papers that I'm a co-author on has published that script. And they basically have to guide you through, like you have the same inputs and stuff like that, but you have to change a few parameters in it. But contact me offline, or maybe I can navigate you to the resource to run that. But you do have to have like Python installed and a few visualization tools. The visualization software that I recommend is Cytoscape for the larger scale. So if you have less than a few thousand genomes, 2, 3, 4, 5,000 genomes. Beyond that, you need actual visualization software like in R, or what did I use for that? I think I used, did I use R for that? I used Gephi. I'm blanking on the name of another, it's a Python package that I used for my very large scale networks. I'll try to think of it and get back to you. But yes, there is a script. I go through the stuff on protocols.io. And if you need any further help, assistance, just, email me, let me know. Oh, okay. Yes, the tutorial will be live. Yes, tutorial will be live. It will be recorded. I will go through most everything. I'll try to spend the first half an hour, 45 minutes going through iVirus, and then another half an hour, 45 minutes through KBase. KBase will appear more polished and presented, but that's literally because I am scrolling through the lab notebook of data. When trimming, we choose quality options for quality threshold, or is it selected automatically? Okay, so this is a great question. I would invite you to look at the protocols.io recommendations for the iVirus stuff. I'll be going through some of this next week. But yes, you should, there are options that you should use for quality control purposes. For Trimmomatic, I'll have to go through them in a little bit more detail. They're automatically assigned for you in iVirus, which is kind of tricky. If you're using it on your local machine or your HPC, you're gonna have to change some of the parameters. But if you're using it on iVirus or KBase, it will automatically be available for you. And what are pros and cons between Galaxy-like platforms and platforms that you showed was my opinion. Yeah, Galaxy is kind of a service that I've used a little bit in the earlier days. I don't feel like it's as, you can do a lot with Galaxy, and there's a lot of apps, a lot of tools and stuff available for you to use on Galaxy. I don't think the resources are as developed as iVirus and KBase in terms of being able to process larger scale data sets and organize them. I also have some issues with the Galaxy interface. And in terms of a developer perspective, I find it a little bit more challenging in a way to use Galaxy. And so, iVirus was really kind of designed or developed to put tools, ready to go tools on platforms that were easier for the developer to develop on, reached a larger community base, and had mechanisms for feedback for the developers and for users. And I'm not knocking Galaxy at all. I think it's great, but I it's maybe a preference. And all the steps that we're going through next week that I talked about this week, most of them you can go through on Galaxy. You'll just have to change some of those options and, fidget around with stuff. Pros and cons, I've answered some of that. And I think next week, for those that you attend next week, I think some of the pros and cons will be apparent when I'm using those platforms. So if you have used Galaxy, I think it will become very clear to you the differences between the platforms. And if not, just ask me again. For viral human database, that is a great question. That is also a great question. I don't do a lot of human work personally. What would I use for a viral data set? I know IMG has a lot of environmental and human data sets. If my boss was like, hey, I need you to do this human viral stuff, RefSeq is inundated with a lot of human viral data sets. I feel like everything from NCBI is human data. So if you compared your data set against NCBI, you'd cover almost all the known human viruses, not the existing human viruses. but most of the known ones. And if you restricted your IMG VR search to human-associated data sets, you'd probably get a lot of really good hits as well there too. But yeah, hopefully that answers it. I hope it's done. Oh, okay. Yeah, I'm pressing yes. Figure out how this answer thing works. Yes. And I'm going to split a couple more. Done. And okay, I think that's it. Thank you everyone for the 16 people that are still here at the end of almost 2 hours of me talking and me losing my voice. Yeah, thank you again. And next week I will also be here to go through the whole pipeline on iVirus and KBase with little pointers for HPC. And I think that's it.